Hi everybody, today we're going to be talking about Mendelian genetics. In the 1800s, people understood that certain characteristics or traits could be bred for. Selective breeding was already in place for dogs or fruits or vegetables, but people didn't really know what DNA was and didn't understand how inheritance worked. They came up with an explanation of why selective breeding worked, and they called it the blending hypothesis. This was a theory where traits from both parents would combine in the offspring. It was almost like blending paint. If you take blue paint and yellow paint, the resulting color will be green. The problem with this hypothesis is that after several generations, everyone should start to look the same. This clearly isn't the case. So another hypothesis was proposed, the particulate hypothesis. This hypothesis thought that selected material from each parent is passed on, but only some of it is expressed in every generation. It's like a deck of cards. You might have an entire deck, but some cards don't show up in every hand. Gregor Mendel was an Austrian monk who studied inheritance in pea plants. His work was published in 1866, but no one took notice of it for almost 40 years. Mendel died in 1884 before he received any accolades for his tremendous contributions to science. Mendel is now known as the father of modern genetics. Mendel observed pea plants for many of his experiments. He looked at the character of the plant. Character is a hereditable feature that can vary amongst individuals. In the example of the pea plants, Mendel was looking at the character of flower color. Traits are the variants of a character. In Mendel's case, he observed plants that had a trait of either purple flowers or white flowers. For Mendel, pea plants were ideal to use because they self-pollinate. The pollen from one flower usually pollinates the other flowers on that same plant. When Mendel's plants self-pollinated, they would produce offspring that had the same character traits as the parent, meaning that all offspring from a self-pollinated plant would look just like the parent. This is called true breeding plant. Mendel started off with stable plants that had been true breeding for generations, always producing the same color flowers in every generation. We've already discussed genotype and phenotype. The genotype is the genetic makeup of the individual, whereas the phenotype is what an individual actually looks like. In true breeding plants, the plants always look like the parents, which means they have the same phenotype. The reason they have the same phenotype is because they have the same genes, or genotype. Let's talk some more about how this works. An allele is a different variant of the same gene. If the character is flower color, and the traits are either purple flowers or white flowers, then there are different alleles that code for purple or white flowers. Here we can see two homologous chromosomes from a diploid cell, and the allele for flower color is highlighted. The top pair has two of the same alleles. This individual would be called homozygous. On the bottom pair, each chromosome has a different allele for flower color. This individual would be called heterozygous. The genotype is which alleles the individual has, whether or not the plant has two of the same or two different alleles. The phenotype is what the flowers actually look like. An individual that is homozygous for the purple allele would have two purple alleles and would have purple flowers. An individual that is homozygous for the white allele would have two white alleles and would have white flowers. In pea plants, the purple allele is dominant. What this means is that an individual that is heterozygous has one purple allele and one white allele, but the purple allele dominates. Therefore, the phenotype is purple. If there is a purple allele present in the cell, the phenotype will always be purple. The other allele, the white allele, is therefore recessive. The only way to get a white flower is to not have a purple allele, to have two white alleles, or be homozygous recessive. If I were to show you this picture and ask you about the phenotype, you could tell me that this plant has white flowers. That is its phenotype. But what if I asked you about the genotype of the plant? You now know that this plant must have two white alleles, as the only way to get a recessive phenotype is to have two recessive alleles. It is homozygous recessive. But what if I showed you this picture and asked what the phenotype is? You would say the phenotype is purple flowers. And what about the genotype? You can't tell me that without further information. A plant that is homozygous dominant for purple alleles will have a purple phenotype, but so will a plant that is heterozygous. The purple plant could have either genotype. When we are discussing alleles and whether they are dominant or recessive, we use a capital letter for the dominant trait. Remember that we are talking about diploid cells here. So you have two homologous chromosomes that each have an allele. For these plants, the purple allele is represented by a capital P. Therefore, a plant that is homozygous dominant and has two purple alleles will have the genotype big P, big P. A plant that is heterozygous will have one dominant purple allele and one recessive allele. It is represented by the genotype big P, little p. And a plant that is homozygous recessive has two recessive alleles will have the genotype little p, little p. Note that the white allele isn't named with a W because it's recessive. It's named with a small p. To review, a plant that is homozygous will have two of the same alleles for a given trait. It can either be two dominant alleles, designated by two capital letters, or a homozygous recessive, designated by two lowercase letters. An organism that is heterozygous has two different alleles, one on each homologous chromosome. This is designated by a big P and a little p. Mendel started out looking at just one trait. He took his true breeding pea plants and bred them. This is called a cross. Since he was looking at only one trait, it is called a monohybrid cross. He then observed the phenotype over multiple generations. The plants he started with were called the P or parental generation. The offspring of that cross is called the F1 generation, and breeding F1 plants together gives you the F2 generation. When we do a cross, we set up a Punnett square, which is a tool that helps us predict the possible genotypic and phenotypic outcomes of a cross. To do a Punnett square, there's a set of steps you can follow to make sure you set it up correctly. We will walk through it together. It consists of determining the genotype of the parents, determining the gametes produced by the parents, setting up the grid, filling in the grid, and then finally assessing the genotypic and phenotypic ratios. The first step in setting up a Punnett square is to determine the genotype of the parent. Mendel already knew the genotype. For multiple generations, the plants with purple flowers always had offspring with purple flowers, so there couldn't have been any white alleles mixed in there. The plants with white flowers, for multiple generations, always had offspring with white flowers, so there couldn't have been any purple alleles in their chromosome. The purple plants are big P, and the white plants are little p, little p. The second step in setting up a Punnett square is to determine the possible gametes that the parents can produce. Think about meiosis. What alleles do each of these true breeding plants have that they can contribute towards making gametes? The purple plants can only produce a gamete with a purple allele, or big P. The white plants can only produce gametes with a white allele, or little p. A Punnett square will give you all of the possible outcomes of a cross. It also gives you useful information about the probability of genotype and phenotype of the offspring. The third step in setting up the Punnett square is to place the gametes from step number 2 along the top and left edge of the grid. To set up the grid, pick one parent to be on the top and one parent to be on the side. It doesn't really matter which is where. I set mine up so that the purple parent is at the top and the white parent is at the side. Place each possible gamete from each parent in its own little box. Step 4 is to fill in the grid. To figure out what goes in this box, you would bring down the gamete from the top and bring over the gamete from the side. By convention, you always place the dominant allele first. So in the first square, you would write big P, little p. Continue through the rest of the boxes, bringing down one allele from the top parent and one allele from the side parent. What we are doing here is simulating fertilization of the gametes. Once you have finished filling in the grid, all of the possible genetic outcomes are represented. Step 5 is to assess the genotypic and phenotypic ratios. In this case, crossing two homozygous parents results in 100% of the F1 generation being heterozygous. The genotype is heterozygous, and the phenotype will be purple. When you observe these plants, they will all look purple. When we cross the F1 generation together, we get an F2 generation, and this gives us much more information about the original genotype of the parent. We go through the steps of the Punnett square again. The first step in creating the Punnett square is to determine the genotype. We already know this. The F1 genotype is big P, little p, which means that all of the F1 generation plants have both big P and little p alleles. The second step in creating the Punnett square is to determine the possible gamete. Since each parent has a genotype of big P, little p, each parent can produce a gamete with either a big P allele or a little p allele. Next, you construct the square with the cross of the F1 generation. We fill in the top and left edges of our square with the possible gametes produced. Filling in the grid would give you this. Our last step is to assess the genotypic and phenotypic ratios. Each square in our Punnett square gives you a possible genotype of the offspring based on independent assortment of the alleles. There is one square where the genotype is homozygous dominant. That is 1 out of 4, or a 25% chance you would get this genotype. Likewise, there is a 1 in 4, or 25% that the offspring will be homozygous recessive. Notice that there are 2 squares where the outcome is heterozygous. That means you have a 2 in 4 chance, or 50% chance, that the offspring of the F1 generation cross will be heterozygous. So your genotypic probability is 25 to 50 to 25%, or 1 to 2 to 1 genotype. If you look at the possible phenotypes, the likelihood of having a big P allele is 3 out of 4, or 75%. All of these plants will have a purple phenotype. The only way to get a white plant is to have a homozygous recessive, and there's only a 1 in 4 chance, or 25% chance of that. So your phenotypic probability is 75% to 25%, or 3 to 1. You can see that the genotype and the phenotype ratios are quite different here. Sometimes you don't know what the genotype is of an organism. Say I gave you a purple plant. You have no idea whether that plant is heterozygous or homozygous dominant. How do you find out? You can do something called a Testcross. This is where you take an organism of unknown genotype and cross it with a homozygous recessive individual. In a Testcross, our unknown plant is purple, which means it can be either homozygous dominant, big P, or heterozygous, big P, little p. The white plant is homozygous recessive, so little p, little p. We follow the same steps for the Punnett square. First, we determine the genotype of the parents. Since we're not totally sure about the purple parent, we're going to have to do a cross for each possibility. Next, we have to determine the possible gametes from each parent. If the purple parent is homozygous, it can only contribute a big P allele. If it's heterozygous, it can contribute either a big P or a little p allele. Let's say the purple plant is the female parent and put its contributions on the side. The white parent's gametes will go on top. Remember, we have to do 2 crosses here because we don't really know what the original purple parent's genotype is. The results from the test cross are striking. If the purple parent is homozygous dominant, you will observe a 100% purple phenotype after the first cross. All of the flowers will be purple. However, if the purple parent is heterozygous, you would observe a phenotypic ratio of 1 to 1. Half of the plants will be purple, half will be white. By doing the test cross, you can determine the original genotype of the purple parent. Mendel discovered that the traits in the offspring of his crosses did not always match the traits in the parental plants. This meant that the pair of alleles in the parental plant had separated or segregated from one another during formation of the gametes. From his data, Mendel proposed the law of segregation. These principles are as follows. A gene can have more than one form or allele, and individuals inherit 2 alleles for any given character. These alleles will separate from one another during gamete formation. We now know that the separation of the 2 alleles occurs during meiosis I, when the homologous chromosomes in the diploid cell are separated to form haploid gametes. Mendel also performed crosses and observed 2 characters at the same time. For example, the color and shape of peas. Since we are tracking 2 traits, you have 4 different alleles to keep track of, as each trait, either color or shape, has 2 alleles associated with it. What Mendel found was that the alleles for the 2 traits will assort independently, but only if they're on different chromosomes. Mendel started with normal, true breeding pea plants, looking at seed color and shape. One of the parents was homozygous dominant for both traits. The other parent was homozygous recessive. In pea plants, round and yellow traits are dominant. So the parent that was homozygous dominant for both traits had a genotype of big Y, big R, which would give a phenotype of round and yellow peas. The other plant was homozygous recessive for both traits. It had a genotype of little y, little r, which gives a phenotype of green and wrinkled peas. We know the genotypes. The second step is to determine the gametes. We have either homozygous dominant plant or a homozygous recessive plant. The homozygous dominant plant can only contribute dominant alleles. The homozygous recessive plant can only contribute recessive alleles. The homozygous dominant plant will make gametes that are dominant for both color and shape, and the homozygous recessive plants will therefore make gametes that contain alleles that are recessive for both color and shape. Our third step is to construct our grid. This one is pretty easy because each gamete only has one possibility for the alleles that it will contain. The F1 generation is going to be 100% heterozygous for both traits. The phenotype is 100% smooth and yellow because those are dominant, and all of the possible offspring have at least one dominant allele. Now we can cross the F1 generation. We know that all of the F1 generation are heterozygous for both traits, so we know the genotype of the parents. Step 2 says we have to determine what gametes can possibly be produced from the F1 parent. Each gamete will have one allele for color and one allele for shape. One possible gamete would be big Y, big R. Alternatively, a gamete could have big Y, little r. A third possibility is little y, big R. And finally, the last possibility is little y, little r. These are the four possibilities of gametes produced from the F1 parents, and this is what you'll be putting into the edges of the Punnett square. We have to produce an extra big Punnett square here because there are four different possibilities of gametes from each parent. Start to make the crosses. The Punnett square shows the possible genotypes of all of the different offspring. Once you know the genotypes, you can start filling in the phenotype. Anything that has at least one big Y and one big R will be yellow and round. You can see there are 9 of these. Anything with at least one big Y but no big R will be yellow and wrinkled. There are 3 of these possibilities. Anything that has no big Y but has a big R will be green and round. And anything that is homozygous recessive, all little y and little r, will be round and wrinkled. For every 16 offspring produced, our phenotypic ratio would be 9 to 3 to 3 to 1. Think about this question. If you did the dihybrid cross and got 160 offspring, how many of those 160 plants would have round and yellow peas? Work on it, and I'll see you in class.